So every great science story begins with science fiction. My story was no different. I remember when I was 7, I finally convinced my parents to let me watch Star Wars. And so I got to the second one, Empire Strikes Back, and there's this scene where Luke Skywalker gets into this battle with the guy who ends up being his father, Darth Vader, and he cuts off his hand. And this could be like really traumatizing for a little kid. Luke Skywalker's walking around without a hand. But next thing you know, Luke's got a new hand, right? It's this robotic engineered hand that's working, and as he thinks about moving this hand is working just like his own. And I remember being struck that picture of technology augmenting and improving folks who had injuries and improving their function never left me. I stayed with that picture until I was finishing undergrad. I did chemical engineering, and I got near the end of undergrad. I was trying to figure out what to do with myself, and all of a sudden I was reading all of these articles showing up in the New York Times about this field that was being launched called brain-machine interface. And I read about the work that was being done at Duke University. It was a pioneering scientist. His name was Miguel Nicolelis. And what he'd figured out how to do was he put individual wires, each the size of a piece of hair, into a monkey's brain. And what he was able to do with these little metal wires was read what the monkey was thinking. So he could get information out of a part of the monkey's brain that dealt with movement, the motor cortex. And so he was seeing the brain as the monkey was moving its arm around. And so he gave the monkey a video game, let's say something like Pac-Man. So the monkey's playing Pac-Man, and he's watching his brain as the monkey's playing Pac-Man. And what they were able to do is take all of these electrical signals that were coming out of the monkey's brain and decode them. And they could say, okay, the monkey's going to move Pac-Man left, I can see left in the brain. The monkey's going to move Pac-Man right, I can see right in the brain. And so they decoded all of these signals and then built a robotic arm. And so instead of sending these robotic signals, right, these signals into another computer, they sent it into the robotic arm. And so as the monkey was playing Pac-Man, moving arm left, moving arm right, the robotic arm started moving around. And then what they taught the monkey how to do was they sent visual feedback to the monkey. So the monkey now realized that he could move this robotic arm. And then the last piece was they disconnected the joystick. So the joystick was no longer feeding into Pac-Man. The monkey's brain was feeding into Pac-Man in the robotic arm. And the monkey learned to use the robotic arm. And there was nothing as powerful as the day when the monkey realized that he didn't actually need to move his own arm to move Pac-Man or the other arm. He could just move the arm by thinking about it. So by thinking about this movement, he could have control over this whole different robotic arm. He'd gotten a new limb. And all of a sudden, this idea that I'd seen as a little kid when I was watching Star Wars, science fiction had become science. I knew I had to go to Duke. I applied and I joined their MD PhD program. And the way it works is you spend 2 years doing all your basic science coursework and your first set of clinical rotations, and then you join the research lab. And so my first clinical rotation, I was stationed at the state psychiatric hospital. And I remember I walked in, I had my little white coat, my clipboard of all the questions I was supposed to ask. And we'd gone through seeing patients and evaluating them over and over again. So I knew exactly what I was supposed to do. I knew what the scene was going to be like. So I was in the state psychiatric hospital. I walk in the room, and I was totally blown away. There was a guy sitting in the corner, and he had a blanket over him, and he was shivering because he was really cold. And I'll paint this scene as in a graphic way as I can. So he had on, the glasses that the doctors give you after you've had your eyes checked, the really thick, dark ones. He had those on. And he was sitting there and he was shivering, and we all have seen little kids when they shiver in the winter and they sort of have a little bit of dripping crystallized on their face. He'd been leaning forward and shivering so long that he'd formed a little crystal on the tip of his nose. And so I walked in and I started asking him, I said, "Sir, so how are you doing? What brought you into the hospital?" And he says, "Man, I'm not doing good. I've got a headache." And I said, "Oh, this is good. Headaches I know how to do. So, sir, so tell me more about this headache. Is it sharp? Is it dull?" And I started running through my medical school checklist. He's like, "Oh, it's sharp." I'm like, "Okay, great. I can do sharp headaches." I said, "Sir, so when did the headache begin?" And he said, "Well, I was back in Vietnam, and the government captured me. And when they captured me, they put my head in a vice. And when they put my head in a vice, they opened it up, and then they inserted a chip into my brain using a drill. And just when the pain stopped, they drilled some more, and then they finally put the chip in there. And ever since they put the chip in my brain, they've been reading all my thoughts, and that's why I have a headache." So all of a sudden, my checklist, I was a little thrown off, as I wasn't quite expecting that. And so I'm thrown off for a minute, and then I think, okay, I've got to go back to this. "Sir, are you seeing things that no one else is seeing?" He says, "No." I said, "Sir, are you hearing things no one else is hearing?" He says, "No." Okay, great. Sir, are you having any thoughts of hurting yourself? Finally, this is the first point in the whole interview. He stops, he pauses, he takes off his glasses, he looks directly at me and he says, you know what, no, I'm not having any suicidal ideations, but I'm starting to have some homicidal ideations. It was time to go. And so after I collected myself and left the room and called my attending, I remember walking out of that experience thinking to myself, you know, we'd gone through so much of pathophysiology, learning about the heart, the lungs, the kidneys, and yet there was a person sitting in front of me. He was clearly sick, and nobody could tell me what was wrong with the organ that was leading to this sickness. And nobody could tell me why the medicines worked or why they didn't work, why this guy had been in the hospital so long, what was the dysfunction that was going on in the organ called the brain. And from that day, I came along this incredible curiosity about wanting to understand how the brain worked, and then asking the question, could we apply technology to ultimately treat folks like the patient that I'd seen that day? And so it was a fascinating, it was an amazing time in science. It was 2003, 2004. They just finished sequencing the human genome. And so there was this massive hope in the field that ultimately we would find the gene for schizophrenia and the gene for depression and the gene for bipolar disorder in the same way that we'd figured out the genes that caused Huntington's disease and the genes that ultimately gave rise to things like cystic fibrosis. And so it was a very exciting time for me. And as things continued and progressed throughout my time in graduate school, it became clearer and clearer that our hopes of finding that single gene probably wasn't what was ultimately going to end up happening. It turned out there were many genes, there were many changes that could happen that could give rise to each of these illnesses. And as I was trying to use technology, to understand how genes changed brain function, it became clear that there was something just was fundamentally missing in the approach and how I was thinking about the problem. So I finished up med school and grad school, and I decided that I just needed to get a better handle around some of the illnesses and that I needed to get more hands-on experience and understand the nuances better. And so I decided to do clinical residency in psychiatry. And there was a patient encounter that I had that shaped how I really see the field evolving and how I see technology ultimately interfacing with health. And so that patient's name was CJ. CJ was a 19-year-old kid, and he graduated valedictorian of his class. He went off to college. He was full of the hopes that he could change the world. But as soon as he got to campus, he started having all these problems concentrating, and they sort of got worse as the semester went along. He failed his first semester of classes, and his parents just had no idea what had happened to their son. They took him to see a psychiatrist and a neurologist and a pediatrician, and after all of this, they had no answers whatsoever. Nobody could tell them what was going on with their son. And the only thing they knew was that CJ was in no condition to go back for a second semester of classes, so they kept him home. And so as they were sitting at the kitchen table one day, CJ, he just gets up, he walks up to his bedroom on the second floor of their house, he opens the window, and he jumps out. And so they're still sitting there at the kitchen table when they heard the crash in the bushes outside. And his parents had no idea what to do. They'd never experienced anything like this before. So they decided that the best thing they could do was just take 12-hour shifts watching CJ. So they took 12-hour shifts, and this went on for like a week and a half. And after that week and a half, they were completely exhausted. And to make matters even worse, CJ wasn't actually getting any better, right? He was now seeing angels and demons. They were talking to him. They were telling him that he was worthless, that he deserved to die. And the pictures on his wall were screaming all day long at him. And they were just completely exhausted. And they knew that they had to finally seek help. So they decided to take him to the hospital. As they were driving on the way to the hospital, CJ reaches over and opens the door in the back of the car and tries to jump out. His mom just happened to be sitting in the back seat with him, so she grabbed his arm and pulled the door shut. And this was CJ's second suicide attempt in the last 2 weeks. The drive, for them, it seemed like an absolute eternity. They finally got to the hospital, and in the hospital, he was in the emergency room. It took him about a week and a half to get admitted, and he finally ended up on my inpatient psychiatric unit. So when I met CJ, he was absolutely terrified. He'd have long conversations with the walls, each one ending the exact same way, with him just begging for them to let him live. I'd seen these symptoms before, and so I started him on an antipsychotic agent. And over the course of several weeks, CJ went from being, what I'd say, it's critically ill to being severely ill. And so I pulled his parents aside to talk to them, and they were at a complete loss, right? CJ's been in the hospital now for multiple weeks, And for them, this wasn't the CJ that they knew, right? They wanted to know what happened with their son. So I studied myself because I'd seen these set of symptoms before. CJ, he had schizophrenia. And so I braced myself because every time I'd have to deliver a diagnosis like this, every family treats it and handles it in a different way. For some people, they're confused. Other people treat it like a death sentence. But CJ's parents, they had a reaction that I'd never quite experienced before. As soon as the words schizophrenia left my mouth, both his parents started trembling. And I thought I needed to explain the disease and the prognosis and the course, and I only got this look of defiance from both of his parents. And so I kept going, and finally his dad just stops and he pauses and he looks at me and he says, Dr. Dzirasa, pick another diagnosis. Pick depression, pick early onset Alzheimer's. He said, pick anything but that. And I thought it was really important to do psychoeducation and help them to understand. And I was absolutely getting nowhere with explaining to them. And finally, CJ's mom looks at me and says, Dr. Dzirasa, you just don't understand. If you give CJ that diagnosis, you don't understand the stigma that he'll have to face. You don't understand the shame that he'll have to deal with for the rest of his life. But I did. My family has struggled with mental illness for generations. In fact, when I was in grad school working on this, all these questions, I had one family member that we had to commit to an inpatient psychiatric unit. It was the first time that my family had ever talked about mental illness. I discovered that one of my parents' siblings, 3 out of 4 of them, carries a diagnosis of schizophrenia or bipolar disorder or depression. I remember what it was like for my family to hire a private investigator. We found one of my family members in the alleyway on another continent hallucinating. So I understand every bit of the stigma and the shame that CJ's parents were dealing with. I understand the stigma. It's so palpable that every time I go off to give a talk, my family members say, just don't let them figure out who I am, right? For me, it was this dramatic transition that happened in grad school, that as I was studying these disorders and there was this tremendous curiosity, all of a sudden this curiosity became fear as I started learning about psychiatric genetics and my family's history. And it was this fear that one day I would wake up hallucinating, or one day I would be seeing demons. And once I transitioned into my 30s and past that critical window, that fear became a fear that one day my children or my nieces or my nephews would be diagnosed or having to deal with one of these illnesses that 20 to 30% of Americans suffer from every year annually. So I started with this dramatic this curiosity about the brain, and that curiosity transitioned into fear of understanding this organ. So the brain is made up over 200 billion cells, and who we are is locked within those cells and their interactions with each other. For centuries, scientists and philosophers have really tried to understand what this organ is and how this organ works. And we've made a lot of progress, so we now understand things like there are differences between your left brain and movement and things like speech and things like creativity. But what was remarkable is that as we were making all that progress, it turned out that those early scientists and philosophers didn't appreciate that your brain used a force that they couldn't see or they couldn't touch. And it turns out that force is electricity. And so I'll frame my talk today from this perspective, and this is a perspective that we've been wrestling with. What if psychiatric illnesses are actually illnesses of electricity? And so this might sound far-fetched, right? I'm a physician, so I can frame these things for you. Imagine if one day you or your friend or your family member was having chest pain, right? You would go to the emergency room. They'd check your blood pressure and your heart rate, and then they would do this strange thing. They would put 12... That was strange. They would put 12 leads on your chest. And those 12 leads, what they would be doing is they would record or measure electrical activity flowing through your heart. And based on those patterns of electricity flowing through your heart, they might make some decisions. On the one hand, they might decide that you just need some medication. On another hand, they might decide that you need a stent or something that opens up blood flow in your heart. But in some cases, what they'll do is put little electrical leads on your chest and deliver a little bit of electricity that allows your heart rhythm to become normal again. In another case, they might say, wow, you need a lot of electricity, and so they'll defibrillate you, right? They'll shock you, your heart back into rhythm. So electricity in this case is in the medicine. And in other cases, one heart rhythm might suggest that they actually need to put in a pacemaker, something that allows you to maintain normal electrical rhythms in your heart. So it's not that far-fetched when we think about the heart that we can have diseases of electricity in the heart that can be treated with electricity. And so it always struck me that it might not be that far-fetched to start thinking about the brain in that manner. All right. So electricity is defined as a movement of charge. In the brain, charge is made up of chemicals. Those chemicals are sodium, potassium, calcium, and chlorine. And just under about half of those 200 billion cells are able to move those chemicals across their first surface to generate electrical pulses. So in the brain, the electrical communication between cells is directly related to chemicals. As psychiatrists, for the last few decades, we've focused on these chemicals, right? It's why we use terms like electrical or chemical imbalances. But for neuroscientists, the real holy grail is understanding how those electrical pulses interact with each other to form feelings and thoughts and emotions. So it turns out if you think about an illness like depression, what most people don't know is that one of the best treatments for depression is actually putting electricity into the brain, directly into the brain. And what surprises many people as I talk about this is that they don't realize that most major medical centers in the country still use this therapy called shock therapy. It's one of the most effective treatments for depression. And so the question then is if the holy grail for neuroscientists and what I would frame as the future of psychiatry is understanding electrical pulses and electrical rhythms and electrical information in the brain, then how come we haven't been able to figure out how this works and come up with new treatments? So I frame this, and I'll frame this from a perspective to say, well, let's say we wanted to understand an electrical machine, like a computer, right? If you wanted to understand a computer, the first thing you might do is take it apart, right? When you're young, it's called breaking things. When you're old, it's called reverse engineering. But the principle is still the same, right? You're going to take it apart piece by piece. So I thought this slide would be a great framing for this idea of taking a computer apart. So this is we'll say the Great Institute of Higher Learning, the Academy of Athens, right? And so this is Plato and Aristotle. They would basically sit around and think about ideas and think about the world and think about how things work. And so the picture's sort of a joke, but you can imagine what would happen if you plopped a laptop computer in the middle of a room one day as these philosophers were sitting around thinking, right? And they probably spend the first, let's say, 7 or 8 hours totally surprised that there was light coming out of this box, right? And they would be staring at it, and they say, what, is like the sun in there? Like, why is there light coming out of this box? But at some point in time, the power would go out, the computer would turn off, and they might take it apart. And as they began to take it apart, they'd see all these cool pieces, right? They'd see like chips, and they'd see hard drives, and they'd see things, but they would have no sort of framework for how these things work together. And even if they were able to sort of piece all the pieces back together, they'd have no concept of like electricity, right? And the role that electricity played in getting this machine to work. And even if they were able to figure out electricity, like they would have no idea like what Word was or PowerPoint was, or we can get into even more complicated concepts like Wi-Fi, or I mean, you could imagine like Plato or Aristotle trying to explain this idea of like a search engine on Wi-Fi, right? You take your best ideas, stick them in the box sends those up into the air sorts all of it out, brings the ideas back in the box, and then gives you the answer, right? This would sound like so incredibly far-fetched. And in a lot of ways, this is where we started with the brain, right? If you look back even 100, 150 years ago, the approach was to take the brain once folks had passed away, and then you do slices of the brain, and you'd look at the pathophysiology or the pathology in the brain. And what you would do is you'd look at someone's symptoms, the problems they were having when they were alive, and then you'd look at their brain when they passed away, right? These are post-mortem studies. And then you try to link the two of these together, right? And there was a lot of progress made in this, right? Folks could have strokes, and you'd see like part of their motor part of their brain was no longer there, and then you'd say, oh, well, the left side of the brain, right side of the body. And then in other cases, you'd have diseases in which people started shaking, and you'd say, oh, well, some of these cells in an area of the brain called substantia nigra have died, and this ultimately became Parkinson's disease. Or you'd look at the cells and you'd look closer and you'd see plaques and tangles, and this became Alzheimer's disease. And so we got understandings of the brain by looking at pathology, right? And these are the diseases, right, which two things happened. One, they generally became named after people, either the people who described the pathology or described the behavioral changes. And so that was the first thing that happened. The second thing is this became an entirely new field called neurology. And this field was the field where there was something wrong with the brain. And then there's an entire different field that emerged, which is when you took the brain and you sliced it up and you did the histology, you didn't see anything wrong. And these became the psychiatric illnesses. And so much of the stigma that we deal with in the context of psychiatric illness is based on this framework of splitting this organ into two forms of diseases, diseases in which there's something wrong with the brain and diseases in which there's something isn't wrong with the brain, right? And this is why psychiatric diseases have names like bipolar disorder or schizophrenia, which means splitting of the soul. It was this inability to link this pathology to an organ. Now, certainly over the course of 100 years, we've learned much more about the brain, including that the receptors, including that what the chemicals are, and we can see changes in those areas, but much of the stigma still persists. But the idea now is that as people started to study the brain, right, so that's the brain when the electricity isn't running through it, as people started to study the brain with the electricity running through it, they noticed some really interesting phenomenon. The first thing that they noticed was that, and this was through putting electrodes, like the heart electrodes, they put them on the head, they noticed these things called brain waves, right, where you would have electricity in the brain and it would sort of go up and down like waves over time. And as they studied these waves, people started having a change in their conceptual framework. And the conceptual framework said, well, what if these waves are like almost like little conductors, right? And they serve as little conductors in an orchestra, and they allow brain cells to keep their tempo, such that brain cells can work together to produce music in the same way that all of the musicians in a symphony can produce music together by being coordinated by the conductor. And as scientists did more and more research around this, what they found in human beings was that many brain cells were actually being coordinated by this brain wave as it went up and down. And they found that as they looked in animals as well, that the same brainwave changes, they can find cells or individual electrical neurons that were being coordinated by these brainwave changes in the brain. So you can see how a brain rhythm or this brain wave or this we'll call it a miniature brain metronome could work. And so I'm just showing a brain wave on the left-hand side, which is setting the tempo. And then every time the brain wave gets to the top, you have a certain number of cells that fire, right? Or they activate or deliver an electrical charge. And then another couple of cells that deliver an electrical charge at the bottom. And this brain wave allows the red cells and the green cells to work together to produce music. And so the question is then fairly simple, right? If there is a brain sort of metronomic system, and if there is, it's important for coordinating the activity of many cells, perhaps psychiatric illness might be due to dysfunction in a brain metronomic system. And this is the framework of the questions that we ask in my lab. Now, what we try to tackle in this case is we use animal models. And the way we frame these animal models is we take animal models and then we expose them to things that give rise to mental illness in humans. And so some of those manipulations might be genes. And so these genes, while not 100% penetrant, can give rise to symptoms that look like things that we see in humans, and I'll talk about that in a second. Or we can expose them to a lot of stress. This stress can be things like separating them from their moms when they're young as a model of childhood trauma. We can expose them to things like bullying in adolescence. Or we can expose them to drugs of abuse, whether it is things like opiates or cocaine or alcohol. And we can see how that changes their brain metronomic systems. And so to get after this, the technology, we're basically using that same sort of technology that was originally used in monkeys to help their brain move. We're now applying that in mice. And so here's what it looks like. We're able to take individual wires, and these individual wires, each the size of a piece of hair, we're able to implant it directly into multiple brain areas simultaneously in a mouse. And that on the bottom, you can see in the blue, there is a CT image. The blue basically probes are wires going into the animal's head. Each one is tinier than a piece of hair, so the mouse can tolerate it exceptionally well. And we can target many brain areas all throughout the depth of the animal's brain. So we're recording many, many, many metronomes and many, many, many cells simultaneously. The animal wakes up from surgery. It's totally fine about 4 or 5 days later. And so as the animal is awake and behaving and moving around and experiencing the world, we're able to measure its brain electrical activity. We're able to get individual cells, so individual neurons firing or activating or delivering these electrical pulses. We can measure them in the animal's brain, and we're able to measure all of these brain waves, or the metronomes, simultaneously as the animal is moving around. Now, this is a massive amount of data, and I want to frame this for you, because I've talked to other collaborators who are doing this type of work around the challenges posed by this. So the amount of data that we're collecting is sometimes on the order of many, many gigabytes, almost terabytes per animal over multiple days. And so the reason why work like this was just not feasible before, I'll sort of start dating myself. When I was in high school, I remember getting my first computer, and it had like 100 megabyte hard drive, right? And I was so excited about it. So 100 million byte hard drive, right? And so now we're collecting 1,000 billion terabytes per day, right? So there was just no way of gathering all of that data into one place, right? The average investigator in the lab couldn't do it. And then the second problem is, once we gather all this data, it's really hard to make all of this data make sense as an animal's running around a cage and interacting with other animals. And so we had to bring in an entirely new field, which is called machine learning. And the idea behind machine learning is it can look through these large, complicated data sets, right, and find patterns. And so we can make these patterns relate to what is going on in the animal's behavior, and then figure out how these patterns are different between the animals that are models of mental illness and then the animals that are normal, so they haven't had the manipulations. Once we're able to do that, then we can figure out where the metronomes are basically dysfunctional in the animal's head, and then we apply stimulation tools to fix that. And so the stimulation tools, we have to have ways of reading information, or reading what the metronomes are supposed to be in the animal's head. We have to pull that information out in real time. That means we've got to be able to do it in about 20 milliseconds. So 20 1/1000th of a second. We've got to be able to get the information out of the animal's head, process what the metronome is supposed to look like, and then send the information back in the animal's head in the right way. And we're able to send that information back in, sending energy back in using either direct electrical stimulation, or we can use light, and this is a technology which if anyone has questions I can tell you about, which uses, you can transform light into energy inside of the brain. So we're either putting electricity back in, or we're putting light back in. And what our early experiments have found is that in these models of mental illness, we can actually normalize their behavior by targeting these individual metronomes. Okay, so I could imagine what everyone is thinking at this point in time. And if you're not thinking it, it is probably what you should be thinking, right? So a mouse doesn't get guilty, right? A mouse doesn't get sad, a mouse doesn't hallucinate, and a mouse certainly doesn't get suicidal. A mouse isn't CJ, a mouse isn't my family member, right? And so I think it's important to say that that isn't exactly the reason why we use mice, right? And I think in the same way that you can study a mouse's electrical rhythms in its heart and figure out things that are principles that are conserved across species, it's the same way that we can use these animal models to figure out how ultimately to target brain pacemakers in humans. So about, wow, time flies. It was about 7 months ago, and I got invited to an event. It was a closing event that was organized by the White House called the Frontiers Conference. And they wanted to talk about the future of brain science and the future of precision medicine. And so I spent about an hour on the stage with the 44th president talking about where we thought technology can go. And he asked a question. There was a question posed to the panel. And the question was, well, what sort of, why haven't we've got all this stuff, right? We've got all these tools. The White House had just launched an initiative 3 years before to create all these tools and technologies to get this type of information out of human beings' head. And the question was, well, why haven't we figured this out yet? And so I thought the best way to do this was to pose a question to the audience. And I asked the question, how many people in the audience were excited about voting? Raise your hand, right? And so people raised their hands, right? You guys are raising your hands. This was obviously much more amusing at the time. It was October 13th. In hindsight, it might not have been that funny. And I described to the president what happened. I explained to him that my mouth generated these pressure waves. The pressure waves traveled through the air, struck the tympanic membrane or the eardrum. that created mechanical force, and mechanical force opened up the hair cells in the ear through the tip links and the kinocilium. It changed that mechanical energy into chemical energy. That chemical energy changed to electrical energy, traveled down the cochlear nucleus into your superior olivary nucleus, into your auditory thalamus, into your auditory cortex, which then became pitch. That pitch, for most people in the room, became words on the left side of their brain. The words became representations. I said if you were young in the room, you probably got excited about voting and released some dopamine from your ventral tegmental area into nucleus accumbens. If you were a little bit more seasoned, you probably remember the first time you voted and your hippocampus became activated. And I said if you're a politician, your amygdala probably lit up as you were counting the number of hands in the room. And then your hand went up, and that entire process happened in half a second, right? And so when we're talking about the brain, we're dealing with this incredibly complex, engineered system, right? We've both got to understand the language of psychiatric illness. We've got to understand how that interacts with electricity. And then we're dealing with all of these parts of the brain that are working together very fast to ultimately produce these changes that we're talking about. But from my perspective, I think the future is bright, right? I'm hopeful. I'm hopeful for my family members. I think ultimately the treatments that we're working on will allow people who've been suffering from illness to come out of the shadows, to play prominent roles in contributing to society and ultimately live their healthiest lives. So we've got about 15 minutes. I'm happy to take any questions if there's anybody in the audience that has any. Thank you. Yeah, no, we haven't, right? So and there are a lot of things that are going to be important for this, right? So there are, and I'll say that the answer would have sounded more like science fiction about 3 months ago, right? But even that's changing. So the first round of technologies that were through this BRAIN initiative, which is the White House's goal of creating new technologies to read information out of human brains, are now implanting electrodes in humans. But the idea of getting this much information simultaneously through all depths of the human brain seemed really far-fetched 7 or 8 years ago. I think we're on the horizon of that. It has not been applied in children. The second challenge is, even if we can get the information out, you've got to figure out how to basically stimulate the brain, right? And there are some tools that are being developed that would allow some to do this. Ultimately, if you can figure out how to do this non-invasively, you'd want to start there, especially with children. There are ways of manipulating magnetic fields outside of the brain to change electrical fields inside of the brain. This is called transcranial magnetic stimulation. And so the idea is to take all of these individual sort of pieces and technologies that we're creating and combine them together in systems that work, and then ultimately we would miniaturize them. The thing that made this seem a lot more like, so when I gave this talk 8 months ago, it sounded like science fiction. In the last 3 months, even Elon Musk has announced launching a company to basically create what is ultimately augmented human intelligence, where there is brain and machine interface, where human brains are interacting with computers in real time.
Is it sustainable when you do these sort of resets in the mice, or is it like a short-term fix?
So some of the things we do can produce what we call long-lasting plastic changes, right? So they can tune the brain to work in a different way. Others, in what we think, so we think different diseases will require different things, right? There are diseases which you get sick and then you get better, and they sort of have this intermittent course, things like bipolar disorder. We think in that case a reset might be okay. There are other diseases in which the neurosystems sort of degrade over time, things like autism and Alzheimer's and schizophrenia, in which the solution will ultimately probably be an implantable pacemaker chip that augments neural systems and allows them to work normally. Yeah. Yeah. Sorry, I'm supposed to wait for the mic to come around.
Great presentation.
Thank you.
My training, I'm a clinical psychologist.
Yeah.
It's fascinating. Piggyback, quick question. Obviously, the mouse and your intervention is very complex, very elegant, very invasionary, the whole deal. And the transcranial stuff, I guess I'm not talking, but it isn't working. At any rate.
Well, I heard you.
Yeah, okay, at any rate. The transcranial approach is still a little primitive, obtuse, whatever word you want to use. Can you speak to that large gap?
Yeah, so I think the technologies are getting better very quickly and are moving together, right? So transcranial magnetic stimulation has been shown to be a fairly effective treatment for depression in some people, right? The idea is, can you get better targeting and can you get deeper? So the problem with transcranial magnetic stimulation is you can't stimulate the deep parts of your brain, right? It stimulates the shallow parts of your brain. People are trying to learn if there's a language they can use, whether it's stimulating two sites simultaneously to cause a certain convergence stimulation deep. It's just an unknown at this point in time. All of that's work that's in progress, and the Brain Initiative has really put a lot of, it was the 21st Century Cures Act that's funded to the tune of a couple $1 billion, put $1 billion over the next 10 years into neuroscience research to do exactly this. So there's a lot of energy around this. Yeah. I'll go back and forth to these sides of the room.
Thank you so much. This has been such an interesting presentation. Do you mind if I ask a philosophical question?
Sure.
You did mention Aristotle. I've read that EEG tests can show in humans that there's activity in the brain's motor cortex fractions of a second before a person consciously decides to act. If that's true, with or without such interventions that you are suggesting are on the horizon, are humans conscious authors of our own decisions?
That's a good one. Yeah, I probably should have paid more attention in that part of psychiatry residency. It's a great question, right? So it depends on how you frame decides to act, right? So if there is a process which initiates a thought of movement, and another process that allows you to become aware of the initiation of the process of movement, and another process that moves, and the timing of those don't line up perfectly, which they don't, right? You might come to the conclusion that our conscious processes don't have anything to do with what we do. I don't think that's the case. Yeah. That seems easier. There we go.
Hi, thank you so much. I'm sure you have maybe thought about, the uses of this technology going forward. You're talking about brain disease, and you mentioned AI. What else do you think this might be used for in the future?
Yeah, so this is complicated, and I think there are a lot of ethical questions that are going to be raised by technology like this, right? And so I've basically been buried in the lab and in the clinic for many years trying to heal my patients and heal my family, right? A lot of the conversation that's happened in the last year, and there's sort of two camps which are pushing this. One camp is it's about augmented intelligence and actually just making human beings smarter for the sake of improving brain function, right? In the same way that we have shoes that allow us to run faster and jump higher, right? So it's this idea of making our function better. There's another arena, which is, and this is an interesting one, it's worried about computers becoming smarter than humans and taking over the world. And the solution to this, the singularity, and the solution to this will be humans integrating with computers because a human computer is better than a computer. Yeah, yep. And so you can read about some of the work being done by Brian Johnson around this. I think we as a society have to wrestle with, like every new technology that comes online, we have to wrestle with like how this impacts who we are. And it's everything from like Facebook on up, right? Technology has a profound impact to change us.
I know the electric shock therapy, does it cause memory loss?
Yes, yeah. So one of the problems with electric shock therapy, right, is you're essentially delivering shock and you're causing the whole brain to seize, right? And so what you're doing is you're actually causing the whole brain to sort of fire together and synchronize itself. So it's almost like this massive reset. So the problem with everything in medicine is the less targeted it is, the more side effects you get. And so the side effect of the electroconvulsive therapy is that some people can have really problematic memory loss. Even though it is the most effective treatment, it has side effects, which is why it's not the most widely utilized treatment.
I was intrigued by your comments early on about your theory that mental illness may be electrical disturbance in the brain. My field is endocrinology, and I have always thought that mental illness, especially depression, bipolar disease, is an endocrine disorder of the brain. Are these two concepts mutually exclusive?
Yeah, no, I don't think it is at all, right? So it's always challenging for some of my neuroscientist colleagues to appreciate that the brain exists within the body and that it it comes to homeostasis with the body. In other words, it balances itself with the rest of the body as well, right? So the brain doesn't like make its own oxygen, it like doesn't make its own glucose, right? It's getting all of these things from the rest of the body. The brain is interacting not only with the endocrine system, it's interacting with the gut, it's interacting with the inflammatory system in a bi-directional way, right? So in other words, the other parts of the body impact the brain, and the brain impacts the other parts of the body, right? So I don't think that that idea is incompatible. It might be a part of what's going on. Now I want to be gentle in how I say this. It could be that there are cases in which the endocrine system interacts with the brain in such a way that it doesn't process electricity correctly. Yeah. It's certainly because you're a doctor, I love answering the questions all the way, right? It is certainly the case that if you have like thyroid dysfunction, you can look like you have depression, right? So, or you can look like you have bipolar disorder. So there's certain things that can happen externally. And the way psychiatrists have dealt with this is if your depressive symptoms are explained by the fact that your thyroid function is low, it is not depression, right? So this is, it's sort of these ways of thinking the way through the brain that are built into our ways we practice medicine that can be really problematic for allowing the whole system, understanding how the whole system works together.
Yeah, I'm sorry. When you described your CJ, many years ago, I saw a patient had the exact same situation, and she dropped out of Harvard, and she ended up having Graves' disease. That was not diagnosed for 6 months before she came to me.
Yeah. Well, one of the patients I had developed autoantibodies against a receptor type in the brain. And so they were basically, their inflammatory system was attacking the cells in the brain that process information, right? So in that case, the fix is not to put pacemakers in the brain, it is to target the immune system, right? But it's the same idea that it's probably breaking the brain's ability to process electricity normally.
With regard to schizophrenia and the increased incidence, or it appears first during teenage years. Have you looked at brain electrical activity in advance of actually young people being symptomatic? And to be able to, where there's either a genetic predisposition or a familial issue, some of the examples you talked about, to assess the likelihood that a particular young person would become more likely to exhibit symptoms, and then to think about what interventions are possible in advance of the person being symptomatic.
Yeah, so I think that is the strategy that we as neuroscientists and psychiatrists should be taking, right? So because I think the heart is such a great framework for this, right? There are certainly interventions that we use for folks after they have had heart attacks, right? We would prefer to give them antihypertensive and anti-cholesterol agents before, right? Because it is a much more effective way, and we can only get the heart function so optimal after the heart attack has occurred, right? So I would think about schizophrenia in the same way. Now, it's not a universal disease. Everybody isn't exactly the same, but there's certainly a decrease in function that can happen with each major psychotic episode. So schizophrenia used to be called dementia praecox, or dementia of the young, right? And so you can think about, and what dementia is, it's a deterioration, a stable, lasting deterioration in function. And so the idea is if we can get on the front side of that, preventing from that from happening, it's a much, much better strategy. And so there's certainly work that's been done on the parts of the National Institutes of Mental Health. They funded this to try to look for signatures of high risk. Some of this includes genetics, some of this includes behavioral changes and family history as well. I think a lot of the work looking at direct brain activity and the processing of brain information is something that they're increasing efforts towards now. So this is where the field is and where we think things are going to evolve. But I think your question is exactly right.
Hi, it was really a fantastic talk. Can I just ask, do you know what happened to CJ?
Yes, yeah. So CJ actually ended up doing much better, right? So CJ was ultimately able to go back to college. In CJ, in some ways, there were some things that worked in CJ's favor that don't always work in everybody's favor. CJ responded well to the medications. So after about 2 months, CJ had finally stabilized and stopped hallucinating. CJ had a super supportive family, which turns out is an incredibly therapeutic part of the process as well. One of the major challenges with mental illness is it attacks your ability to advocate for yourself, right? So I always describe these as illnesses that affect families. And CJ had great care very early on in the diagnosis, right? So if you think about a disorder that creates dementia, the longer you wait and the longer you go, the harder it is to sort of recover normal function. And so CJ was ultimately able to go back to school and get a job.
Very interesting talk. My question to you is, what is the status of developing signatures for schizophrenia, whether it's electronic or fMRI, and do they correspond with the psychiatric definitions that are out there?
This is a fantastic question. So the status is this is literally the cutting edge, right? So the BRAIN Initiative launched in 2013. The DARPA, this is the defense agency that ultimately gave us the internet, put about their initial $80 million into this. And so the studies have funded and were initiated in the last 3 years trying to look for these exact same signatures. One of the approaches and the frameworks that the last director of the National Institutes of Mental Health took, and the new one took, is part of the reason we haven't done a great job of linking like psychiatric diseases to biology is because our diagnoses might be wrong, right? And so the way we define disease categories might be so all over the place that they don't actually mirror biology. And so this is the history of psychiatry, right? Our disease categories are used so that when I see something and I explain it to the next psychiatrist, we sort of understand that it's a common thing that we're seeing, but it doesn't mean it matches biology. And the way I would explain that is if you called the illness fever, right? So let's say we have an illness called fever. And a patient comes in, they have fever, I see a fever, you see a fever, we measure it, the temperature's high. And I want to do a study of the biology of fever, and I want to test a group, and I give them antibiotics, and some people get better off their fever, and some people don't, right? And so it means the biology doesn't match the disease in most cases. It's because it doesn't, right? Antibiotics don't make you better unless you have a bacterial illness, and it depends on what bacteria you have. And if you have a fever, the antibiotics aren't going to make you better. If you have a heat stroke, the antibiotics aren't going to make you better. So I think in a lot of ways the challenge that we have as a field is we have these disease categories that are big, and the biology is very distinct within these large disease categories. Last question. I got the last question slide.
So I remember in the early 1980s as a pre-med student plastering electrodes on schizophrenics' brains to measure, I think it was the P50 wave. Yeah, so this idea of the brain waves and schizophrenia and how that connects has been going on for a long time. But so I'm particularly interested, one, in what mouse model you use for schizophrenia, because it's so hard to reproduce. And then I'm also thinking about if you make advances and you're actually trying to use some kind of therapy affecting the brainwaves for schizophrenics, about how you will talk to people about that who so often have the paranoia that you described, that someone's putting a chip in their brain, and it sounds really challenging.
It's what actually started with my first story of my encounter with a patient with schizophrenia, talking about them having a chip in their brain, right? So there are, so what she was asking about is this idea that people would do studies, they put electrodes or EEGs on the brain, and then they measure how the brain filters out information, right? And the premise here is that in individuals that have schizophrenia, their brains do not filter out background noise as well as other individuals do. And so that sort of framework, the challenge with that is we're not sure if EEG is sensitive enough, right? So it's like measuring the surface of a ball without actually seeing what's going on inside. And so that's one of the first round of evolutions that have changed. In terms of our mouse models, we use a lot, right? And our premise there is if we can find common biology that converges across a lot of models, then that is our best approach to it. So genetic models, including disrupted in schizophrenia 1 and 22q11, which give risk for these things in humans. We can give things like PCP, which make human beings hallucinate, and ketamine. And so we're essentially looking at convergence points behind that, right? But our premise in our framing, and this is to go back to your question, is that we think the pacemakers are ultimately going to target things like attention and social systems and information storage, right, rather than schizophrenia. And so if we can frame these models and see how these genes or these manipulations alter a working memory system, then we are generating a working memory pacemaker that's useful for schizophrenia and Alzheimer's. So that's the framework. Yeah. Thank you all.